In today's class, we are going to revise the important MCQ points in chapter Cell, the unit of life. The first point is centriole. It forms the basal body of cilia, flagella and spindle fibers. So centrioles. In addition to this, I will mention few points. Centrioles are present in animal cells, but they are absent in plant cells. It forms the basal body of cilia, flagella and spindle fibers. So centriole forms the basal body of cilia and flagella and spindle fibers. They are present in the animal cells, but they are absent in plant cells. Now second point is cristae. They are nothing but infoldings of mitochondria. You have mitochondria inside the cell. Mitochondria is present in the cytoplasm. This is called as the powerhouse of the cell because ATP is generated inside mitochondria. This mitochondria is a double membranous structure that is made up of two membranes. The outer membrane, this is the outer membrane, and you have one more membrane, inner membrane, but this inner membrane is folded like this, which gives a wrinkled appearance. This foldings of the inner membrane towards the matrix is called as cristae. This foldings is called as cristae. This is one more MCQ point. What is the function of cristae? It increases the surface area. Plus, it contains various enzymes responsible for metabolic reaction. Responsible for metabolic reaction. Chlorophyll. Third point is chlorophyll. This is a green pigment which helps in capturing it traps the light energy. Which is used for photosynthesis. Okay, so this chlorophyll is a green pigment which is present inside the thylakoids. These thylakoids are present inside the chloroplast of the plant cell. This function of this chlorophyll is it is a green pigment. It helps in capturing the light energy which is helpful in photosynthesis. Next you have ribozymes. This one you have to understand, this is not ribosomes, these are ribozymes. So ribozymes are nucleic acids. Some of the nucleic acids act like enzymes. Enzymes means which helps in biochemical reaction. RNA acting. Ribozyme means the RNA. Ribozymes are RNA, ribonucleic acid, which acts as enzymes. These enzymes, what are enzymes? They are the biological catalyst. To speed up the biological reactions. So these ribosomes are ribonucleic acids which acts as an enzymes. These enzymes acts as a biological catalyst and helps to speed up the biological reaction. So centriole forms the basal body of cilia and flagella and spindle fibers. They are present in animal cells but absent in plant cells. Cristae, they are the infoldings of mitochondria. Functions of cristae is to increase the surface area and it contains variety of enzymes which is used for metabolic reaction. Chlorophyll, this is a green pigment which is present in the thylakoids. These thylakoids are present inside the chloroplast of the plant cell. They help in capturing the light energy which is used in photosynthesis. Ribozymes, these are ribonucleic acids which acts as enzymes. The function of enzymes is to speed up the biochemical reactions taking place inside the cell. Structures which perform the function of mitochondria. In bacteria, you call it as mesosomes. So, these mesosomes are equal to mitochondria in bacteria. Mesosomes, they are present in bacteria. They perform the function of mitochondria, that is generation of energy. These mesosomes are produced by infoldings of plasma membrane present in the bacteria. So mesosomes carry out respiration in bacteria. These structures, the mesosomes carry out respiration in bacteria. Osmotic expansion of a cell kept in water is chiefly regulated by vacuoles. So other important points about vacuoles are their single membranous structure. That is, it has got only one membrane. They are present in the cytoplasm. These vacuoles are present in the plant cell or large. They help in maintaining the osmotic expansion of the cell. That is, these vacuoles stores water and macromolecules such as ions, sugars, proteins. All these things are present inside the vacuole. So these vacuoles store the excess water and salt and other ionic substances inside, which is responsible for creating the osmotic pressure inside the cell. So osmotic expansion of a cell kept in water is chiefly regulated by vacuoles. The bacteria, that is the motile bacteria. Motile means the bacteria which is able to move. They are able to move because of the presence of structure called as flagella. This is made up of flagellin, a protein called as flagellin. Flagella helps in locomotion, that is movement of bacteria from one place to another place. Microtubules are the constituents of spindle fibers, centrioles and cilia. Mitochondria and chloroplast are semi-autonomous organelles. Autonomous means it can operate on its own. Protoplast is a cell without a cell wall. If you take off the cell wall from the cell, then you call it as protoplast. Ribosomes, they are not membrane bound structures, that is the structures which are not covered by a membrane inside the cell or ribosomes. Functions of gap junction is to facilitate communication between adjoining cells by connecting with the cytoplasm. So through the cytoplasm, two cells get connected with the help of gap junctions. It helps in rapid transfer of ions, small molecules and some large molecules. The important structure, that is the Golgi complex, it plays a major role. That is the function of Golgi complex is post-translational modification of proteins and glycosidation of lipids. This is going to be a quick revision, so I am not going to explain it in detail. A major site for synthesis of lipids is smooth endoplasmic reticulum. We have got two types of endoplasmic reticulum. One is smooth type and another one is rough endoplasmic reticulum. This smooth endoplasmic reticulum is responsible for the production of lipids. Rough endoplasmic reticulum is responsible for protein synthesis. Ribosomal RNA is actively synthesized in nucleolus. A structure which is an organelle within an organelle. You have to understand a structure which is an organelle within an organelle is ribosomes. The green pigment chlorophyll is present inside thylakoids. These thylakoids are flattened membranous sacs forming the grana of chloroplast. Cytochromes are found in the cristae of mitochondria. DNA is present in three organelles: nuclei, mitochondria and chloroplast. Nuclear envelope is derived from, that is nuclear envelope is formed from rough endoplasmic reticulum. The structures that are formed by stacking of organized membranous stacks in the chloroplast. Stacking means adding, suppose you arrange coins like this, one on top of the other. Okay, so the structures that are formed by stacking of flattened membranous stacks. Flattened means like this, on both sides it will be flat. This is a membranous stack which is present in the chloroplast. You call it as grana. The chromosomes in which centromere is situated close to one end is called as acrocentric. Inclusion bodies found in prokaryotes are cyanophacian granule, glycogen granule and phosphate granule. Smooth endoplasmic reticulum is responsible for synthesis of lipids. Eukaryotic cells, examples are euglena, amoeba and paramecium. E. coli is a prokaryote. These are all direct questions, so it doesn't need any explanation. In eubacteria, a cellular component that resembles eukaryotic cells is plasma membrane. This explains the similarity between eukaryotic cell and eubacteria. They both contain only one thing which is similar, that is the plasma membrane. Small cells have a larger surface area per volume ratio. Phagocytosis and pinocytosis are collectively termed as endocytosis. Vital stains are employed to study the living cells, not the dead cells. Vital stains are helpful in studying living cells. Outside the primary wall, middle lamella is present. Fluid mosaic model was given by Singer and Nicholson. Plasma membrane is made up of lipids, proteins and carbohydrates. Middle lamella is composed of calcium pectate. Genophore means either DNA or an RNA which is present in the prokaryote. In prokaryotic cells, these are the special features of prokaryotic cells, that is prokaryotic cells do not have nucleus, 70s ribosomes is present, internal compartments are absent. Plant cells do not have centrioles. Chlorophyll in chloroplast is located in grana. The chemiosmotic theory of ATP synthesis in the chloroplast and mitochondria is based on the proton gradient. The main organelle involved in modification and routing of newly synthesized proteins is to the destination is endoplasmic reticulum. In chloroplast, chlorophyll is present in the thylakoids. The non-membranous organelles, that is the organelles which do not contain membrane or which do not covered by membrane or centrioles, ribosomes and nucleolus. The organelles which contain only one membrane are lysosomes, glyoxysomes and spirosomes. Ribosomes that are present in mitochondria are exclusively 50S type. Powerhouse of the cell are mitochondria. An organism exclusively with 70 type of ribosomes contain circular naked DNA. Inner membrane of mitochondria forms crystal. F1 particles present in the mitochondria are oxysomes. Golgi complex works for secretion. Spindle fibers are formed by microtubules. Acrosome, this is also an important question. Acrosome is formed by Golgi apparatus. Structural lipids of cell membrane are phospholipids. That is, the lipids of cell membrane are phospholipids. Glycocalyx is associated with plasma membrane. A structure which is an effective transport pathway between two adjacent cells, that is between two cells. The mode of transport is by means of plasmodesmata. The main arena of various types of cellular activities. The main arena means the place where all the important activities of the cell takes place is cytoplasm. Cell theory does not apply for virus. Site of formation of glycoproteins and glycolipids is Golgi apparatus. Both the glycoproteins and glycolipids is Golgi apparatus. The difference between rough endoplasmic reticulum and smooth endoplasmic reticulum is rough endoplasmic reticulum contains ribosomes on the surface of endoplasmic reticulum. Elioplast store fats. Golgi apparatus modifies and packages proteins.